So here's something worth thinking about tonight. Right now, as you're lying there, settling in, getting comfortable, every single cell in your body is reading instructions. Billions of tiny molecular machines are crawling along strands of a molecule so thin you couldn't see it with the most powerful light microscope for most of human history. And they're reading a code that was written. Well, that's the thing. Nobody wrote it exactly. It assembled itself, letter by letter, over roughly 4 billion years of trial and error, and the version running inside you right now is the latest draft of the most successful instruction manual ever produced. It tells your cells how to build you, how to keep you alive, how to fight off infections and digest your dinner and fire the neurons that are processing my voice right now. And the whole thing fits inside a space so small that if you unraveled all the DNA from just one of your cells and stretched it out straight, it would be about 6 feet long, roughly your height. But it's packed into a nucleus that's about 6/millionths of a meter across. That's like taking a thread that stretches from New York to Los Angeles and somehow coiling it neatly into something the size of a basketball. And that's just one cell. You have roughly 37 trillion of them. But we're getting ahead of ourselves. Let's start at the beginning. Not the beginning of DNA. We'll get there. But the beginning of people figuring out that DNA even existed. Because for most of human history, nobody had the faintest idea what was going on inside a living cell. They didn't even know cells existed. Think about what people understood about heredity before modern science. They knew, obviously, that children looked like their parents. Farmers had been selectively breeding animals and crops for thousands of years. The ancient Greeks had theories. Hippocrates thought that every part of the body produced tiny seeds that somehow combined during reproduction. Aristotle disagreed. He thought the male provided a kind of formative principle and the female provided the raw material. They argued about it. People argued about it for centuries, actually, and nobody could resolve it because nobody could see what was happening at the scale where it actually mattered. The first real crack in the mystery came in 1869, and it came from a place you might not expect: used surgical bandages. There was a young Swiss physician named Friedrich Miescher. He was 25 years old, working in a laboratory in Tübingen, Germany, in the castle, literally a medieval castle that had been converted into lab space. Miescher was interested in the chemistry of cells, specifically white blood cells, and the easiest way to get large quantities of white blood cells in the 1860s was from the pus on discarded surgical bandages. I know, not glamorous, but Miescher was methodical and curious, and he started breaking open those cells with various chemical treatments, trying to isolate and identify the different substances inside them. He found the usual things: proteins, lipids, what you'd expect. But then, he found something else. Something in the nuclei of the cells that didn't behave like any known protein. It was rich in phosphorus, which was unusual. It was acidic. It resisted the enzymes that normally broke down proteins. Miescher called it nuclein because it came from the nucleus. And here's the thing. He had just discovered DNA. He was literally holding the molecule of heredity in a test tube, and he had no idea what it did. Nobody did. It would take another 84 years before anyone figured that out. Miescher published his findings in 1871, and the scientific world mostly shrugged. Nuclein was interesting, sure, but proteins were the stars of the show. Proteins were complex, diverse, capable of doing all sorts of things inside cells. Most scientists assumed that whatever carried hereditary information had to be a protein, because what else could be sophisticated enough to encode the staggering complexity of life, this assumption that proteins were the important molecules and nuclein was just some structural filler would persist for decades and send researchers down countless dead ends. It's a great example of how preconceptions can blind even brilliant people. But let's back up for a second, because while Miescher was scraping pus off bandages in Tübingen, something else was happening that would turn out to be enormously important. About 500 miles to the southeast, in a quiet monastery in Brno, which was then part of the Austro-Hungarian Empire, now the Czech Republic, an Augustinian friar named Gregor Mendel had already done something remarkable. Between 1856 and 1863, Mendel had grown and carefully crossbred nearly 30,000 pea plants in the monastery garden. He tracked specific traits across generations: flower color, seed shape, plant height. And he discovered something that no one had ever clearly demonstrated before. Heredity followed mathematical rules. Mendel showed that traits were passed from parents to offspring in discrete units, not blended together like mixing paint, which was the prevailing theory, but inherited as distinct particles that maintained their identity across generations. A plant could carry a factor for purple flowers and a factor for white flowers, and the purple might dominate in one generation, but the white factor was still there, hidden, waiting to reappear in the next generation in a precise, predictable ratio. 3-to-1, roughly. Purple to white, tall to short, round seeds to wrinkled seeds, over and over the same ratios. This was revolutionary. Mendel had discovered what we now call genes, though he didn't use that word, and he'd figured out the basic rules of how they're transmitted. He presented his findings to the local Natural History Society in 1865 and published them in 1866. And then, almost nothing happened. His paper was cited about 3 times in the next 35 years. The scientific establishment largely ignored him. Mendel himself seemed to sense that he was ahead of his time. He reportedly told a colleague, "My time will come." He died in 1884, 16 years before it did. Now, here's what makes this doubly poignant. Mendel and Miescher were working at almost exactly the same time, on 2 halves of the same puzzle. Mendel had discovered the rules of heredity, the abstract logic of how information passes between generations. Miescher had discovered the physical substance that carries that information. But neither of them knew the other's work was relevant, and nobody connected the dots for decades. It's one of those moments in the history of science that makes you want to reach back through time and introduce people to each other. The connection started to form around the turn of the 20th century. In 1900, 3 different botanists, Hugo de Vries in the Netherlands, Karl Correns in Germany, and Erich von Tschermak in Austria, independently rediscovered Mendel's laws. They'd each been doing their own breeding experiments, gotten similar results, and then found Mendel's paper gathering dust in the literature. Suddenly, Mendel's time had come, just as he'd predicted. The scientific world woke up to the idea that heredity was particulate, carried in discrete units that followed mathematical rules. But what were those units, physically? Where did they live inside the cell? This is where a German biologist named Theodore Boveri and an American graduate student named Walter Sutton come into the story, working independently around 1902 and 1903. They both noticed something striking: the behavior of chromosomes during cell division, the way they paired up, separated, and distributed themselves into daughter cells, looked an awful lot like the behavior Mendel had described for his hereditary factors. Chromosomes came in pairs. Mendel's factors came in pairs. During the formation of reproductive cells, chromosome pairs separated so each egg or sperm got only one copy. That's exactly what Mendel's model predicted for his factors. The parallel was too perfect to be coincidence. This became known as the chromosome theory of inheritance, the idea that Mendel's abstract hereditary factors were physically located on chromosomes. And it was controversial. Not everyone bought it right away. But it pointed scientists in the right direction, toward the nucleus, toward the chromosomes, toward the physical stuff of heredity. Now chromosomes were visible under a microscope. Scientists could stain them, watch them move during cell division, count them. And they knew that chromosomes were made of two main components, protein and that nuclein stuff Miescher had found, which by now was being called nucleic acid, specifically deoxyribonucleic acid, or DNA. The question was, which one carried the genetic information, the protein, or the DNA? And almost everyone bet on protein. It made intuitive sense. Proteins are made from 20 different amino acids, which gives them enormous combinatorial complexity. You can arrange 20 different building blocks in a staggering number of ways. DNA, on the other hand, seemed boringly simple. It was made from just 4 chemical building blocks, 4 nucleotide bases. How could a molecule with only 4 letters in its alphabet possibly encode the instructions for something as complex as a human being? It seemed absurd. The great biochemist Phoebus Levine even proposed what he called the tetranucleotide hypothesis, suggesting that the 4 bases in DNA just repeated in a simple, monotonous pattern, a CGT, ACGT, ACGT, over and over. If that were true, DNA couldn't carry meaningful information any more than a string of ABCD, ABCD, ABCD could encode a novel. So DNA was dismissed as structural scaffolding, and proteins got all the attention. This went on for years, decades really, and it took a series of ingenious experiments to overturn it. The first major crack came from a British medical officer named Frederick Griffith, working in London in 1928. Griffith was studying pneumococcus bacteria, the kind that cause pneumonia, and he was trying to develop a vaccine. He was working with 2 strains of the bacterium. One strain was virulent. It had a smooth polysaccharide capsule that protected it from the immune system and could kill mice. The other strain was harmless. It lacked the capsule and was easily destroyed by the immune system. Griffith called them the S strain for smooth and the R strain for rough, based on how their colonies looked on a petri dish. Here's what Griffith did, and here's where it gets genuinely eerie. He took the deadly S strain and killed it with heat, dead bacteria. Then he mixed those dead S bacteria with living R bacteria, the harmless ones, and injected the mixture into mice. Now neither the dead S bacteria nor the living R bacteria should have been able to harm the mice. The S bacteria were dead. The R bacteria were harmless, but the mice died. And when Griffith examined their blood, he found living S bacteria, smooth-capsuled, virulent, deadly. Somehow, something from the dead S bacteria had transformed the living R bacteria, giving them the ability to make the capsule, turning them lethal. Griffith called this the transforming principle. Something, some substance, had passed from the dead cells to the living ones, carrying with it the instructions for making a polysaccharide capsule. It was as if the dead bacteria had bequeathed their genetic information to the living ones. But what was that substance? Griffith didn't know. He died in 1941, killed when a German bomb hit his laboratory during the London Blitz, and he never found out. But someone else picked up the thread. A quiet, meticulous Canadian-American researcher named Oswald Avery, working at the Rockefeller Institute in New York City. Avery was in his 60s by the time he zeroed in on the transforming principle, and he'd spent most of his career studying pneumococcus. He was cautious by nature, almost painfully careful, the kind of scientist who wouldn't publish until he was absolutely certain. And starting in the late 1930s, he and his colleagues Colin MacLeod and Macklin McCarty set out to identify exactly what the transforming principle was. Their approach was elegant in its logic. They took extracts from the S strain bacteria and systematically destroyed one type of molecule at a time, then tested whether the extract could still transform our bacteria. They used enzymes that specifically broke down proteins. Transformation still worked. They used enzymes that broke down RNA. Still worked. They used enzymes that broke down lipids. Still worked. But when they used an enzyme that specifically broke down DNA, a DNA SE, transformation stopped cold. The transforming principle was DNA. Avery published these results in 1944, and you might think the scientific world would have immediately recognized this as one of the most important discoveries of the 20th century. But it didn't play out that way. Many scientists remained skeptical. Maybe the DNA preparation was contaminated with trace amounts of protein, they said. Maybe the real transforming agent was a protein too small to detect. The protein hypothesis had so much momentum, so much institutional weight behind it, that even strong evidence couldn't dislodge it overnight. Avery himself was cautious in his claims, almost frustratingly so. He never won a Nobel Prize for this work, which is widely considered one of the great oversights in the history of the Nobel Committee. But the evidence kept building. In 1952, Alfred Hershey and Martha Chase at Cold Spring Harbor Laboratory on Long Island performed an experiment that was harder to argue with. They used bacteriophages, viruses that infect bacteria, and they asked a beautifully simple question. When a virus infects a bacterium, what actually goes inside? The virus's protein coat, or its DNA, because whatever goes inside is carrying the genetic instructions that hijack the cell. They used radioactive labeling to track each component separately. They tagged the protein with radioactive sulfur, because protein contains sulfur and DNA doesn't. They tagged the DNA with radioactive phosphorus, because DNA contains phosphorus and protein doesn't. Then, they let the labeled viruses infect bacteria, and they used a kitchen blender, literally a wearing blender, to shake the empty viral coats off the outside of the bacteria. When they checked where the radioactivity ended up, the answer was clear. The phosphorus, the DNA, was inside the bacteria, directing the infection. The sulfur, the protein, was outside, discarded like an empty syringe. DNA was the genetic material. By the early 1950s, the evidence was becoming overwhelming, and the race was on to figure out what this molecule actually looked like, what its structure was, because if you could understand the structure, you might understand how it worked, how it stored information, how it copied itself, how it did the thing that life requires above all else, which is to reproduce faithfully, generation after generation, while still allowing for the occasional variation that lets evolution do its work. And that race, the race to solve the structure of DNA, would become one of the most famous, most dramatic, and in some ways most troubling stories in the history of science. It involved a cast of characters you couldn't make up if you tried. A brash young American who'd gotten his PhD at 23, a talkative British physicist who'd been told by his boss to work on something else, a brilliant and fiercely independent woman whose data would prove essential and whose contribution would be scandalously overlooked, and a quiet New Zealand-born chemist who almost beat them all to it. They were all converging on the same question in the early 1950s, working in labs scattered across England. and what happened next would change everything about how we understand life itself. So let's talk about these people. Let's start with the quiet one, because his role in this story is complicated, and he tends to get lost in the noise. Maurice Wilkins was born in New Zealand in 1916, grew up in England, studied physics at Cambridge, and during the Second World War, he worked on the Manhattan Project, specifically on isotope separation for the bomb. And like a lot of physicists who worked on that project, the experience left him deeply unsettled. He came out of the war wanting to do something that felt more life-giving, more constructive, and he found himself drawn to biophysics. This idea that you could use the tools of physics to understand living systems. He ended up at King's College London in a lab run by John Randall, and by the late 1940s, he'd started working on DNA. Specifically, he was trying to use X-ray diffraction to figure out the molecule's structure. Now, X-ray diffraction. Let's take a moment with this, because it's going to be absolutely central to everything that happens next. The basic idea is beautifully simple, even if the execution is fiendishly difficult. You take a beam of X-rays and you aim it at a crystal or a fiber of the molecule you're interested in. The X-rays hit the atoms in that molecule and scatter. They bounce off in different directions, and the pattern they make when they hit a piece of photographic film on the other side tells you something about how those atoms are arranged. It's a bit like shining a flashlight through a complicated piece of lace and then trying to figure out the pattern of the lace by studying the shadows it casts on the wall behind it. Except the shadows are made of X-rays. The lace is a molecule, and the math required to work backward from the pattern to the structure is extraordinarily demanding. The technique had been around since the early 1900s. The Braggs, William Henry Bragg and his son, William Lawrence Bragg, a father-son team who shared a Nobel Prize in 1915, had pioneered it for simple crystals, things like table salt. By the 1930s and 1940s, people were using it on bigger and more complex molecules. Dorothy Hodgkin, one of the great crystallographers of the 20th century, used it to solve the structures of penicillin and later vitamin B12. Linus Pauling, over at Caltech, had used it along with his extraordinary chemical intuition to figure out key structures in proteins, including the alpha helix, a corkscrew shape that turns out to be one of the fundamental building blocks of protein architecture. Pauling published that result in 1951, and it sent shockwaves through the field because it showed that you could figure out the three-dimensional shape of a biological molecule, and that the shape would tell you something profound about how the molecule worked. So Wilkins was trying to do the same thing with DNA. He'd gotten some good fibers of DNA. He was getting some preliminary X-ray images, and things were looking promising. And then something happened that would poison the atmosphere at King's College for years. John Randall, the lab director, hired a new researcher, Rosalind Franklin, and assigned her to the DNA project. But the way Randall communicated this to Franklin and to Wilkins created a disastrous misunderstanding. Franklin arrived at King's in January 1951, believing that the DNA work was hers, her project, her problem to solve. Wilkins, who'd been away when she started, came back expecting her to be his collaborator, maybe even his assistant. Neither of them was wrong about what they'd been told. Randall had simply been unclear, or perhaps deliberately ambiguous, and the result was a working relationship that was strained from the very first day and never recovered. Here's the thing about Rosalind Franklin. She was, by any measure, an extraordinary scientist. She'd been born in 1920 into a prominent London family, studied chemistry at Cambridge, where women could attend lectures and sit exams but couldn't actually receive degrees until 1948, which tells you something about the world she was navigating. After the war, she'd spent several years in Paris at a government research lab where she'd become genuinely expert in X-ray diffraction techniques applied to carbon and coal. She was meticulous, rigorous, and she had an almost uncanny ability to extract structural information from X-ray photographs. She was also, by many accounts, reserved and private, someone who didn't suffer fools and didn't engage in the kind of casual, speculative banter that characterized a lot of the scientific culture at Cambridge. At King's College, which in the early 1950s was a fairly conservative, male-dominated, almost clubby environment, the senior common room where researchers ate lunch didn't even admit women. She was isolated from the start. And the tension with Wilkins made everything worse. But here's what matters most. Franklin was brilliant at what she did, and she immediately began producing X-ray diffraction images of DNA that were better than anything anyone had seen before. She figured out that you needed to carefully control the humidity of the DNA fibers, and she discovered something crucial, that DNA existed in two forms. At lower humidity, you got what she called the A form, which produced a very detailed but complicated diffraction pattern. At higher humidity, the fibers absorbed water and stretched out into what she called the B form, which gave a simpler, more striking pattern. And it was the B form that would turn out to be the key to everything. Now let's cross the English Channel. Well, actually, let's just go up the road to Cambridge, about 60 miles north of London, because that's where the other half of this story was unfolding. The Cavendish Laboratory at Cambridge was one of the most storied physics labs in the world. This was where J.J. Thomson had discovered the electron, where Rutherford had split the atom, where Chadwick had found the neutron. By the early 1950s, it was run by Lawrence Bragg, the same Bragg who'd shared the Nobel Prize with his father for X-ray crystallography back in 1915. And working in the Cavendish, supposedly on other things, were two men who could not have been more different from each other in background, but who turned out to be perfectly matched in temperament and obsession. Francis Crick was 35 years old in 1951, which made him old for someone who hadn't yet finished his PhD. He'd started in physics, had his graduate work interrupted by the war. He'd designed mines for the Royal Navy, and afterward had drifted into biology, convinced that the big unsolved problems were there. He was tall, loud, laughed constantly, and had a habit of telling other scientists what was wrong with their work, which did not always endear him to his colleagues. Bragg, his supervisor, found him exasperating. But Crick had something genuinely rare. He could think across disciplines. He understood physics. He understood chemistry. He was teaching himself biology, and he had an almost intuitive feel for how molecular structures worked in 3 dimensions. He could look at a set of constraints. This bond angle has to be this, that distance has to be that, and somehow see the shape that satisfied all of them at once. James Watson arrived at the Cavendish in the fall of 1951. He was 23, American, and he'd come to Cambridge on a postdoctoral fellowship that was supposed to be about studying the biochemistry of viruses. But Watson had attended a conference in Naples earlier that year, where he'd seen Maurice Wilkins present some of his preliminary X-ray images of DNA, and something had clicked. Watson became convinced that the structure of DNA was the most important unsolved problem in biology, and he wanted to be the one to solve it. He was ambitious in a way that was almost startling, not in a corporate, ladder-climbing way, but in the way of someone who genuinely believed that solving this one problem would be the most important thing anyone could do. and who saw no reason why he shouldn't be the one to do it. He was also, by his own later admission, not particularly skilled in crystallography or chemistry. What he had was drive, a nose for the right problem, and the extraordinary good fortune to end up sharing an office with Francis Crick. They hit it off immediately. Watson brought the biological urgency, the conviction that DNA's structure was the prize worth chasing. Crick brought the structural thinking, the physics, the ability to reason from first principles about how atoms arrange themselves. And they both brought a willingness to speculate, to build models, to guess and check and guess again. That was very different from the painstaking, data-first approach that Franklin favored down at King's College. And this difference in style, this philosophical gap between model building and data gathering, is one of the things that makes this story so rich and so uncomfortable, because both approaches have real value. Franklin believed, with good reason, that you should collect the best possible data first and let the structure emerge from the evidence. Watson and Crick believed, also with good reason, that you could use physical and chemical constraints to narrow down the possibilities and then check your model against the data. In the end, both approaches were necessary, but the way they came together involved a breach of trust that has been debated ever since. Their first attempt was a disaster, and it's worth telling because it shows you how science actually works, messily, with wrong turns and embarrassments. In November 1951, Watson traveled down to King's College to attend a seminar where Franklin presented some of her recent results on DNA's structure. He didn't take notes. He tried to remember the key numbers she mentioned, things like the water content of the fibers and certain dimensions from the diffraction patterns, and when he got back to Cambridge, he and Crick immediately started building a physical model. They used metal rods and clamps and connectors, the same kind of model-building kit that Pauling had used so successfully for the protein alpha helix. Within days, they had a structure. A 3-stranded helix with the sugar phosphate backbone running down the middle and the bases, those 4 chemical letters, adenine, thymine, guanine, and cytosine, pointing outward. They were so excited that they invited the King's College team up to Cambridge to see it. Wilkins came, and Franklin came, and Franklin took one look at the model and told them it was wrong. The water content was off. Watson had misremembered her numbers. And having the backbone on the inside with the bases pointing out didn't match her data at all. She was right, and she was not gentle about it. The model was dismantled. Bragg, embarrassed, told Watson and Crick to stop working on DNA and go back to their actual assigned projects. And for a while, they did. Or at least they appeared to. But the obsession didn't go away. They kept thinking about it, kept talking about it over lunch, kept reading everything they could find. And meanwhile, down at King's College, Franklin was quietly producing the single most important piece of experimental evidence in the entire story. A photograph so clear, so striking, that when you hear what it showed, you'll understand why it changed everything about the race to understand DNA's structure. It's called Photograph 51, and it's one of the most famous images in the history of science. Not because it's beautiful in any conventional sense. It's a pattern of dark smudges and spots on a piece of film, the kind of thing you'd walk right past if it were pinned to a bulletin board. But if you knew how to read it, if you understood the language of X-ray diffraction, it was screaming the answer. So let's talk about what Franklin actually did. Remember, X-ray diffraction works by firing a beam of X-rays at a crystalline or semi-crystalline sample, and then capturing the pattern that emerges on the other side as those X-rays scatter off the atoms in the structure. The pattern you get is not a photograph of the molecule. That's a really important thing to understand. It's more like a shadow, but a shadow cast in a strange mathematical space. The arrangement of spots and streaks tells you about the spacing and symmetry of the repeating units inside the molecule. But you have to work backward through some pretty serious mathematics to translate that pattern into an actual three-dimensional structure. It's like hearing an echo in a canyon and trying to figure out the exact shape of the canyon walls from nothing but the sound. That's the kind of problem Franklin was solving, and she was extraordinarily good at it. She had trained in Paris under Jacques Meringue, one of the best crystallographers in the world, and she had a meticulous, patient approach to her work that was perfectly suited to this kind of painstaking analysis. She didn't guess, she didn't speculate, she collected data, and she let the data speak. By early 1952, Franklin had figured out something that nobody else had clearly established, that DNA existed in two distinct forms depending on how much water was present. She called them the A form and the B form. The A form was drier, more crystalline, and produced very complex diffraction patterns that were hard to interpret. The B form was wetter, more like a fiber, and it produced something much simpler and more striking. And on May 2nd, 1952, her graduate student Raymond Gosling, working under her direction, took an X-ray diffraction photograph of the B form of DNA that came out with stunning clarity. That's photograph 51. Here's what it showed. The pattern was an X shape, a clear, unmistakable cross of dark streaks radiating outward from the center of the image. And if you know anything about diffraction theory, an X-shaped pattern is the signature, the fingerprint, of a helix. It's what you get when X-rays scatter off a structure that spirals. The angle of the X tells you the pitch of the helix, how far it goes before completing one full turn. The spacing of the spots along the arms of the X tells you the distance between repeating units, and there was a crucial absence in the pattern too. The fourth layer line was missing, which told you something about the symmetry of the structure. Franklin noted all of this. She was working through the mathematics carefully, systematically. She wasn't rushing to publish a model. She was building the case, layer by layer. But here's where the human drama gets complicated, and honestly, a little painful. Because Franklin didn't know that her photograph was about to be shown to someone she hadn't authorized to see it. In January 1953, Wilkins showed photograph 51 to James Watson. The circumstances are still debated in their details, but the broad outline is clear. Watson had come down to King's College, and Wilkins, frustrated by his difficult working relationship with Franklin and eager to share what he saw as exciting progress, pulled out the photograph and showed it to him. Watson didn't have Franklin's permission. Franklin didn't even know it had happened. And Watson, the moment he saw that X-shaped pattern, immediately recognized what it meant. He later wrote that his pulse began to race, that the cross pattern was unmistakably the signature of a helical structure. He didn't need to do the detailed math. The visual pattern alone told him that DNA was a helix, and it gave him crucial dimensions to work with. Now, let's be clear about something. Watson and Crick didn't steal Franklin's data in the way that word is sometimes used. There was no break-in, no cloak-and-dagger espionage. But they did gain access to her experimental results without her knowledge or consent, and those results were absolutely critical to their success. It wasn't just photograph 51 either. Around the same time, Max Perutz, Crick's supervisor at the Cavendish, shared with Crick a Medical Research Council report that contained Franklin's detailed measurements of the unit cell dimensions of DNA. This report included her finding that the phosphate groups were on the outside of the structure, not the inside. Remember, that was exactly the mistake Watson and Crick had made in their first failed model. They'd put the backbone on the inside. Franklin's data told them, definitively, that the backbone was on the outside and the bases pointed inward. So now Watson and Crick had two absolutely essential pieces of information that they hadn't generated themselves. They knew DNA was a helix, and they knew the backbone was on the outside. And with those constraints in hand, they went back to model building. But they still had a massive puzzle to solve. How did the bases fit together on the inside of the helix? There are 4 bases: adenine, thymine, guanine, and cytosine, and they come in varying amounts in different organisms. This is where another piece of the puzzle comes in, one that had been sitting in the published literature for a few years, waiting for someone to recognize its significance. An Austrian-born biochemist named Erwin Chargaff had been doing careful chemical analyses of DNA from different species throughout the late 1940s. And he'd found something peculiar. In every organism he tested, whether it was yeast or wheat germ or human tissue or bacteria, the amount of adenine was always roughly equal to the amount of thymine, and the amount of guanine was always roughly equal to the amount of cytosine. Always. A equals T, G equals C. These became known as Chargaff's rules, and when Chargaff actually met Watson and Crick in the summer of 1952, he was not impressed by them. He later described Watson as someone who didn't know the basic chemistry of the bases, and he found Crick brash and overconfident. Chargaff himself didn't grasp the structural implications of his own ratios. He saw them as an interesting chemical regularity, not as a clue to the architecture of the molecule. But Watson and Crick, once they were back in model building mode in early 1953, began to see that those 1-to-1 ratios might be the key to everything. Here's the thing. If adenine always pairs with thymine and guanine always pairs with cytosine, then maybe the 2 strands of the helix are held together by specific pairings between the bases. Maybe adenine on one strand always faces thymine on the other, and guanine always faces cytosine. And maybe that's what holds the whole thing together. Watson started playing with cardboard cutouts of the bases at his desk, trying to figure out how they could pair up. At first, he tried pairing like with like, adenine with adenine, thymine with thymine, but that didn't work because the pairs came out different sizes, and the helix would have bulges and pinches instead of smooth, regular geometry. Then Jerry Donahue, an American crystallographer who happened to share their office, pointed out that Watson was using the wrong chemical forms of the bases. The textbooks Watson had been consulting showed the bases in what's called their enol form, but Donahue told him the bases almost certainly existed in their keto form inside the cell. This might sound like a minor technical detail, but it completely changed the geometry of how the bases could hydrogen bond to each other. And when Watson switched to the keto forms and tried pairing adenine with thymine and guanine with cytosine, each unlike pair, something magical happened. The pairs came out exactly the same size. An adenine-thymine pair was the same width as a guanine-cytosine pair. That meant you could slot any combination of base pairs into the helix, and the overall diameter would stay perfectly constant. The structure would be smooth, regular, and elegant. Think about what that means. It means the 2 strands of the helix are complementary. If you know the sequence of bases on one strand, you automatically know the sequence on the other. If one strand reads ATGC, the other strand must read TACG. The information on one strand is a mirror image of the information on the other, and that immediately, breathtakingly, suggests how the molecule could copy itself. You pull the two strands apart, and each strand serves as a template for building a new partner. Every A attracts a new T, every C attracts a new G, and you end up with two identical copies of the original double helix. The structure doesn't just store genetic information, it contains, within its own architecture, the mechanism for its own replication. Crick reportedly walked into the Eagle pub in Cambridge on February 28, 1953, and announced that they had found the secret of life. And for once, that kind of grandiose claim was not really an exaggeration. They published their paper in the April 25, 1953 issue of Nature. It was astonishingly short, just over one page, about 900 words. The famous opening line is beautifully understated. We wish to suggest a structure for the salt of deoxyribose nucleic acid. This structure has novel features which are of considerable biological interest. Considerable biological interest. That might be the greatest understatement in the history of science. And near the end of the paper, they included one more perfectly crafted sentence. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. They knew exactly what they had. But here's what's important to sit with for a moment. That same issue of Nature contained two other papers, one by Wilkins, Stokes, and Wilson, and one by Franklin and Gosling, that presented the X-ray diffraction evidence supporting the Watson-Crick model. Franklin's paper included data from photograph 51. The three papers were published together, giving the impression of a collaborative effort. But the reality behind the scenes was far more complicated. Franklin, by this point, had actually left King's College. She'd moved to Birkbeck College to work on the structure of tobacco mosaic virus, and she never fully knew the extent to which her unpublished data had informed Watson and Crick's model. And this is where the story takes on a weight that goes beyond science. Rosalind Franklin died of ovarian cancer on April 16, 1958. She was 37 years old. The cancer may well have been caused or accelerated by her extensive exposure to X-rays during her research, the same X-rays that produced photograph 51 and all those other diffraction images. She worked almost until the end, publishing 17 papers in her final 5 years, doing brilliant work on virus structures. She never won a Nobel Prize because the Nobel is not awarded posthumously, and when Watson, Crick, and Wilkins shared the Nobel Prize in Physiology or Medicine in 1962, she was not alive to be considered. Watson's 1968 memoir, The Double Helix, portrayed Franklin in ways that many scientists found dismissive and sexist, focusing on her appearance, characterizing her as difficult and territorial, failing to acknowledge how essential her data had been to their discovery. It took decades and the work of later historians, especially Brenda Maddox's biography published in 2002, to restore a fuller and fairer picture of Franklin's contributions. So that's the discovery of the double helix, one of the most consequential moments in the history of biology, and also one of the most human, tangled with ambition and rivalry and missed credit and untimely death. But knowing the structure was only the beginning, because now the question shifted. You have this beautiful double helix with its paired bases, A with T and G with C, and you know it somehow encodes the instructions for building a living organism. But how? How does a sequence of 4 chemical letters translate into the proteins that actually do the work inside your cells? The enzymes that digest your food, the hemoglobin that carries oxygen in your blood, the collagen that holds your skin together. That's the problem that consumed molecular biology for the next decade, and the story of how it was solved involves a completely different cast of characters, a different kind of ingenuity, and one of the most elegant logical puzzles in all of science, because someone had to figure out the code, the actual rules by which DNA's 4-letter alphabet gets translated into the 20-letter alphabet of proteins. And the person who first cracked that problem open wasn't working in a famous lab at Cambridge or Caltech. He was a physicist with a sense of humor and a love of puzzles, and his name was George Gamow. And Gamow was, honestly, one of the most colorful characters in 20th century science. Born in Odessa in 1904, he'd already had a remarkable career in physics before he ever thought about DNA. He was one of the people who helped explain how radioactive decay works through quantum tunneling. He'd done important work on the Big Bang theory. He was actually one of the first to predict the cosmic microwave background radiation that was later discovered. And he had this irrepressible, playful personality. He loved jokes, loved riddles, loved games. He was the kind of physicist who would send letters to colleagues written in code, just for fun. And in 1953, when he read Watson and Crick's paper in Nature, something clicked in his mind. He looked at that double helix with its 4 bases and he thought, this is a coding problem. This is cryptography, and I know how to think about codes. Here's the puzzle Gamow was staring at. You've got DNA, which uses an alphabet of 4 letters, A, T, G, and C. And you've got proteins, which are chains of amino acids, and there are 20 different amino acids used by living things. So somehow, sequences of those 4 DNA letters have to specify which amino acid goes where in a protein. The question is, what's the code? How many DNA letters do you need to specify one amino acid? Think about it like this. If each single letter coded for one amino acid, you'd only be able to specify 4 amino acids. A means one thing, T means another, G means a third, C means a fourth. But there are 20 amino acids, so one letter isn't enough. What about pairs? 2-letter combinations of 4 bases give you 4 times 4, which is 16 possibilities. Still not enough. You need at least 20, but 3-letter combinations? 4 times 4 times 4 gives you 64. That's more than enough. 64 possible 3-letter codes for just 20 amino acids. So Gamow proposed, quite logically, that the genetic code was a triplet code, groups of 3 bases, which he called codons, each specifying 1 amino acid. Now he got a lot of the details wrong. His specific model involved overlapping codes, where the same base could be part of multiple codons, and he proposed that the code was read directly from the DNA double helix itself, with amino acids fitting into diamond-shaped holes between the base pairs. That turned out to be completely incorrect. But the core insight, that the code is a triplet code, was absolutely right, and it framed the entire problem for everyone who came after him. And Gamow did something else that was really clever. In 1954, he founded what he called the RNA Tie Club, a group of exactly 20 members, one for each amino acid, who would correspond and share ideas about cracking the genetic code. Each member got a tie with a little RNA helix embroidered on it, and each was assigned an amino acid as a nickname. Watson was proline. Crick was tyrosine. It was whimsical and informal, but it created this incredible network of thinkers all focused on the same problem. Some of the most important ideas in molecular biology were first circulated as informal notes among Tie Club members. But here's the thing. Before anyone could crack the code, they had to figure out something more basic. How does the information in DNA actually get to where proteins are made? Because proteins aren't assembled in the nucleus where DNA lives. They're built out in the cytoplasm, on little molecular machines called ribosomes. So there had to be some kind of messenger carrying the instructions from DNA to the ribosome. And this is where RNA comes back into the story in a big way. Now, scientists had known about RNA for a while. It's chemically similar to DNA, a nucleic acid with a sugar phosphate backbone and bases hanging off it, but with a few key differences. RNA uses the sugar ribose instead of deoxyribose. It's usually single-stranded instead of double-stranded, and it uses the base uracil instead of thymine. So where DNA has A, T, G, and C, RNA has A, U, G, and C, but the pairing rules still work. A pairs with U in RNA, just like A pairs with T in DNA. Through the 1950s, several researchers noticed that cells actively making lots of protein also had lots of RNA, and much of that RNA seemed to be associated with ribosomes. But the picture was confused for a while because there are actually several different kinds of RNA doing different jobs, and it took time to sort them out. The key insight came in 1960 and 1961 from several directions at once. François Jacob and Jacques Monod at the Pasteur Institute in Paris had been doing brilliant work on gene regulation in bacteria, how cells turn genes on and off, and they proposed the existence of a short-lived RNA molecule that carried the message from a gene to the ribosome. They called it messenger RNA, or mRNA. Around the same time, Sidney Brenner and François Jacob, working with Matthew Meselson at Caltech, did an elegant experiment showing that this messenger RNA existed, that it was a transient copy of the gene that traveled to the ribosome and directed protein synthesis. And Crick, ever the theorist, had actually predicted something like this a few years earlier. So the picture was coming together. DNA sits in the nucleus, holding the master copy of the instructions. When a gene needs to be expressed, when the cell needs a particular protein, that stretch of DNA gets copied into a messenger RNA molecule in a process called transcription. The mRNA then travels to a ribosome, and there the sequence of bases in the mRNA gets read in triplets, each triplet specifying an amino acid, and the protein gets built one amino acid at a time. That reading process is called translation. Transcription and translation, DNA to RNA to protein. Crick called this the central dogma of molecular biology, and while the name is a bit grand, and Crick himself later admitted he'd used the word dogma without fully appreciating its meaning, the idea has held up remarkably well. Information flows from DNA to RNA to protein, not perfectly, not without exceptions, but as a general rule, that's how it works in every living cell on Earth. But there was still a missing piece. You've got the mRNA arriving at the ribosome with its sequence of codons, and you need to somehow match each codon to the right amino acid. How does the ribosome know that, say, the codon GCU means alanine? There has to be an adapter, something that can read the codon on one end and carry the correct amino acid on the other end. And Crick, in a remarkably prescient 1955 paper, predicted exactly this. He called it the adapter hypothesis. He proposed that there must be small molecules that serve as translators between the language of nucleic acids and the language of amino acids. And sure enough, those adapters were found. They're called transfer RNA or tRNA. Each tRNA molecule is a small RNA that folds into a distinctive cloverleaf shape. On one end, it has an anticodon, a 3-base sequence that pairs with a complementary codon on the mRNA. On the other end, it carries the appropriate amino acid. So when the ribosome is reading along the mRNA and it encounters, say, the codon GCU, a tRNA with the anticodon CGA comes in, and that tRNA is carrying alanine. The amino acid gets added to the growing protein chain, the ribosome moves forward 3 bases to the next codon, and the process repeats. It's like a molecular assembly line, and it's happening right now, billions of times over, inside your body. Let that sink in for a moment. Every cell in your body contains roughly the same DNA, the same master blueprint. And right now, as you're lying here, those cells are selectively reading different parts of that blueprint, transcribing specific genes into mRNA, and translating those messages into proteins, all with astonishing speed and accuracy. A ribosome can add about 20 amino acids per second to a growing protein chain, and your cells contain millions of ribosomes all working simultaneously. The sheer scale of it is staggering. But we're getting ahead of ourselves a little because the actual cracking of the code, figuring out which codons specify which amino acids, that's a story worth telling properly. And it starts with a young, relatively unknown biochemist named Marshall Nirenberg. Nirenberg was working at the National Institutes of Health in Bethesda, Maryland in the early 1960s. He wasn't part of the RNA Tie Club. He wasn't at one of the famous labs. He was, by most accounts, a quiet and methodical researcher, and he was working with a German postdoc named Heinrich Mattei. Together, they devised an experiment that was breathtakingly simple in concept. They created a cell-free system. Basically, they broke open bacterial cells and extracted all the molecular machinery needed for protein synthesis, the ribosomes and tRNAs and enzymes, everything except the DNA. Then they added synthetic RNA of known composition and watched what proteins came out. Their first experiment, in May 1961, used an RNA molecule made entirely of uracil, poly-U, just U, over and over. They added this to their cell-free system and waited to see what protein would be produced. And what they got was a protein made entirely of the amino acid phenylalanine, polyphenylalanine, which meant that the codon UUU specifies phenylalanine. That was the first word of the genetic code to be deciphered. Nirenberg presented these results at the International Congress of Biochemistry in Moscow in August 1961, and at first, almost nobody came to his talk. He was a nobody, scheduled in a small session. But Crick heard about it, and Crick immediately recognized how important it was, and he arranged for Nirenberg to present again to a larger audience. And this time, the room was packed. The response was electric. People understood instantly that this was the key to cracking the whole code. What followed was an intense race. Nirenberg's lab kept working with synthetic RNAs of different compositions, and so did Severo Ochoa's lab at New York University. Ochoa had actually developed the enzyme used to make synthetic RNA and had won a Nobel Prize for it. There was real competition, sometimes tense competition, between the two groups. But the synthetic RNA approach had limitations. It could tell you the composition of a codon, but not the exact sequence. Knowing that a codon containing 2 U's and 1 C codes for leucine doesn't tell you whether it's UUC, UCU, or CUU. The sequencing problem was solved through additional clever techniques. Nirenberg and his colleague Philip Leiter developed a method using short synthetic RNA molecules just 3 nucleotides long, actual triplets, and testing which amino acid-carrying tRNA would bind to a ribosome in response to each triplet. Meanwhile, Har Gobind Khurana at the University of Wisconsin developed methods to synthesize RNA with precisely defined repeating sequences, which allowed him to confirm codon assignments through a completely different approach. Between Nirenberg, Khurana, and their colleagues, by 1966 the entire genetic code was cracked. All 64 codons were assigned. 61 of them code for amino acids, and 3, UAA, UAG, and UGA, are stop signals that tell the ribosome to release the finished protein. And here's what's really beautiful about the code. It's almost universal. With only minor variations, the same codons specify the same amino acids in virtually every living organism on Earth. In bacteria, in oak trees, in mushrooms, in whales, in you. The code that tells a bacterium how to build its proteins is essentially the same code that tells your cells how to build yours. That's one of the most profound pieces of evidence for the common ancestry of all life. The code was established very early in the history of life, probably close to 4 billion years ago, and it's been conserved ever since. It works, and evolution doesn't mess with things that work at such a fundamental level. Nirenberg and Khurana shared the Nobel Prize in Physiology or Medicine in 1968, along with Robert Hawley, who had determined the complete structure of a tRNA molecule. And there's something satisfying about Nirenberg's story. This quiet, persistent scientist who wasn't part of the inner circle, who didn't have the pedigree or the connections, but who asked the right question and designed the right experiment and broke the problem wide open. Now, knowing the code was a triumph, but it immediately raised new questions. Because the code is redundant, 61 codons for only 20 amino acids means that most amino acids are specified by more than one codon. Leucine, for example, has 6 different codons, and that redundancy has a pattern to it. If you look at the codons for a given amino acid, they usually differ only in the third position. The first 2 bases are the same, but the third can vary. Crick noticed this and proposed what he called the wobble hypothesis, the idea that the pairing between the third base of the codon and the first base of the anticodon is less strict, allowing some flexibility. This means a single tRNA can sometimes recognize more than one codon, which is elegant and efficient because it means the cell doesn't need 61 different tRNAs. But the genetic code, as gorgeous as it is really just the dictionary. It tells you what the words mean. The deeper question, the one that would consume molecular biology for decades to come, is how the cell decides which words to read and when. Because every cell in your body has the same DNA, the same complete set of instructions. A liver cell and a neuron and a skin cell all carry identical genomes. So what makes them different? The answer is gene regulation, the intricate system of controls that determines which genes are turned on, which are turned off, and how much protein each active gene produces. And the first people to really crack that problem were 2 French scientists working at the Pasteur Institute in Paris, François Jacob and Jacques Monod. And their story is one of the most beautiful in all of biology, because they figured out something genuinely profound using nothing more than bacteria, clever genetics, and relentless logic. Jacob and Monod were studying E. coli, that workhorse bacterium that keeps showing up in our story, and they were interested in a seemingly mundane question. How does E. coli decide whether to digest lactose? See, bacteria are opportunists. They'll eat whatever sugar is available, but they prefer glucose. It's easier to metabolize, requires fewer enzymes, and gives them energy faster. Lactose, on the other hand, requires a special enzyme called beta-galactosidase to break it apart into glucose and galactose before the cell can use it. Making that enzyme costs energy. So if there's no lactose around, why bother? The bacterium shouldn't waste resources building a protein it doesn't need. And indeed, that's exactly what happens. When you grow E. coli in a medium with only glucose, the cells produce almost no beta-galactosidase. But the moment you add lactose and remove the glucose, the cells start churning out beta-galactosidase within minutes. Hundreds, even thousands of molecules per cell. It's like flipping a switch, and when the lactose runs out, production stops just as abruptly. Now, think about what that means. The gene for beta-galactosidase is always there in the DNA. It doesn't appear and disappear. The cell's genome hasn't changed. What's changed is whether that gene is being read, whether it's being transcribed into messenger RNA and then translated into protein. Something is controlling access to that gene, turning it on and off in response to the environment. And Jacob and Monod wanted to know what that something was. They spent years on this through the late 1950s, and what they eventually proposed in 1961 was a model so elegant it won them the Nobel Prize in 1965. They called it the operon model. Here's how it works, and I want you to picture this, because it's really satisfying once you see the logic. Imagine the stretch of DNA that contains the gene for beta-galactosidase. Actually, there are 3 genes clustered together that are all needed for lactose metabolism. They sit right next to each other on the chromosome, and they're all transcribed together as one long messenger RNA. Jacob and Monod called this cluster of co-regulated genes an operon, specifically the lac operon, for lactose. Now, just upstream of these 3 genes, there's a short stretch of DNA called the operator. It's not a gene itself. It doesn't code for any protein. It's a control region, a landing pad. And there's another gene nearby, separate from the operon, that codes for a protein called the repressor. Here's the key. The repressor protein, when it's produced, physically binds to the operator. It sits right on top of it, like a parked car blocking a driveway. And when the repressor is sitting on the operator, RNA polymerase, the enzyme that reads genes and makes mRNA, can't get past. It's physically blocked. The genes downstream are silenced. No mRNA is made, no beta-galactosidase is produced. So the default state is off. The repressor keeps the gene shut down. But here's what's clever. When lactose enters the cell, a small derivative of lactose called allolactose binds to the repressor protein and changes its shape. It's like a key fitting into a lock on the side of that parked car, causing it to roll out of the way. The repressor, with allolactose attached, can no longer grip the operator. It falls off. And now, RNA polymerase can cruise right through, transcribe the genes, and the cell starts making the enzymes it needs to digest lactose. When the lactose is used up, there's no more allolactose to distract the repressor. The repressor snaps back into its original shape, rebinds the operator, and shuts everything down again. The whole system is self-regulating. It's a feedback loop, controlled entirely by the presence or absence of the very substance the genes are designed to handle. And this is where you start to see DNA, not just as a blueprint, but as something more like a dynamic, responsive operating system. The information is all there, but the cell has sophisticated machinery to decide which parts of that information to access at any given moment. Jacob and Monod had discovered the first genetic switch. Let that sink in for a moment. Every cell in your body, and there are roughly 37 trillion of them, carries the same DNA, the same complete instruction manual, But a red blood cell reads the chapter on hemoglobin and ignores the chapter on digestive enzymes. A pancreatic cell reads the chapter on insulin and skips the chapter on keratin. A neuron reads entirely different chapters still. Gene regulation is the reason you're not just an undifferentiated blob of identical cells. It's the reason you have a body with specialized tissues and organs that do different things. And it all comes down to which genes are switched on and which are switched off, in which cells, at which times. Now the ELAC operon is a relatively simple system, one repressor, one operator, a handful of genes. But it opened the floodgates. Once biologists understood that gene regulation was a real, concrete, mechanistic phenomenon, not some vague hand-waving about cellular destiny, they started looking for regulatory systems everywhere. And they found them. Layers upon layers of them. In bacteria, the operon model explains a lot. But in more complex organisms, in you and me, gene regulation turns out to be staggeringly more elaborate. And this is where the story gets really interesting, because the complexity of regulation in human cells is honestly almost incomprehensible in its intricacy. Let's talk about what makes it so much more complicated. First, your DNA isn't just floating freely in the cell the way bacterial DNA is. In your cells, DNA is packaged tightly. Remember, if you stretched out all the DNA in a single human cell, it would be about 2 meters long. 6 feet of molecular thread that has to fit inside a nucleus that's roughly 6 micrometers across. That's like stuffing 25 miles of thread into a tennis ball. So the DNA is wound around small proteins called histones, like thread around tiny spools. 8 histone proteins form a core, and the DNA wraps around it about 1 3/4 times, creating a structure called a nucleosome. Then the nucleosomes themselves are stacked and coiled into higher-order structures, compacting the DNA further and further until you get the dense, visible chromosomes you might remember from biology class. But here's the thing. That packaging isn't just about fitting everything in. It's also a form of regulation. When DNA is tightly wound around histones, the genes in that region are essentially inaccessible. RNA polymerase can't reach them. They're silenced, not because a repressor is sitting on them, but because they're physically buried. To activate those genes, the cell has to loosen the packaging, chemically modify the histones so the DNA unspools a bit, making those genes available for transcription. And the way the cell does this is through chemical tags. Small chemical groups, acetyl groups, methyl groups, phosphate groups, can be added to or removed from the histone proteins. Acetylation of histones, for instance, generally loosens the grip between the histone and the DNA, opening up the chromatin and allowing gene expression. Deacetylation does the opposite, tightens things up, shuts genes down. Methylation can go either way depending on where exactly the methyl group is placed. This system of chemical modifications is sometimes called the histone code, and it adds an entire layer of regulation on top of the DNA sequence itself. The genes haven't changed. The A's, T's, G's, and C's are all the same. But the accessibility of those genes, whether they can be read or not, is controlled by these chemical decorations on the proteins that package them. And this is part of what's known as epigenetics, literally, above genetics. Information that influences gene expression without altering the underlying DNA sequence. And it gets even more layered than that. Beyond histone modifications, the DNA itself can be chemically tagged. In many organisms, including humans, methyl groups can be added directly to cytosine bases in the DNA, particularly when a cytosine sits next to a guanine, what's called a CPG site. When a gene's promoter region, the stretch of DNA where RNA polymerase initially binds to begin transcription, is heavily methylated, that gene is typically silenced. The methyl groups physically interfere with the transcription machinery, and they also recruit additional proteins that further compact the chromatin. What's remarkable is that these epigenetic marks can be inherited when a cell divides. When DNA is replicated, the methylation pattern on the original strand is recognized by maintenance enzymes that add matching methyl groups to the new strand. So a liver cell divides and produces two liver cells, not because the DNA has changed, but because the epigenetic marks, the pattern of which genes are accessible and which are locked away, is faithfully copied along with the DNA itself. The cell remembers what kind of cell it is. So now you've got multiple layers of gene regulation working simultaneously. You've got transcription factors, proteins that bind to specific DNA sequences near genes and either help recruit RNA polymerase or block it, similar in spirit to Jacob and Monod's repressor, but far more varied and numerous. The human genome codes for roughly 1,600 different transcription factors, and they work in combinations. A given gene might require 3 or 4 specific transcription factors to all be present and bound before it can be activated. Change one factor, and the gene stays silent. This combinatorial logic is how a limited number of regulatory proteins can control tens of thousands of genes in precise, cell-type specific patterns. Then you've got enhancers, regulatory DNA sequences that can be thousands or even millions of base pairs away from the gene they control. They work because DNA, remember, is flexible. It can loop back on itself, bringing a distant enhancer into physical contact with a gene's promoter. Picture a long, floppy piece of string. You can easily pinch 2 distant points together. That's what happens in 3-dimensional space inside the nucleus. An enhancer far upstream or even downstream of a gene can loop around and touch the promoter, delivering transcription factors and co-activators right where they're needed. And then there are the silencers, insulators, non-coding RNAs, chromatin remodeling complexes, layer after layer of regulatory machinery, all working in concert to ensure that the right genes are expressed in the right cells at the right time and in the right amounts. It's a symphony, and the genome is both the score and the orchestra. Here's something that might surprise you. Only about 1.5% to 2% of the human genome actually codes for proteins. For a long time, the rest was dismissively called junk DNA, the assumption being that it was evolutionary debris, leftover sequences with no function. But as our understanding of gene regulation has deepened, it's become clear that a huge portion of that so-called junk is actually regulatory. Enhancers, silencers, promoters, sequences that are transcribed into non-coding RNAs that themselves regulate other genes. The regulatory architecture of the genome may actually be more complex than the protein coding genes it controls. And this brings us to something that was quietly revolutionizing molecular biology while the world was still celebrating the cracking of the genetic code. Because once you understand that genes can be regulated, turned on and off, dialed up and dialed down, you start to wonder about the mistakes. What happens when regulation goes wrong? What happens when a gene that should be silent gets switched on at the wrong time, or a gene that should be active gets permanently shut down? And the answer, in many cases, is disease. And not just any disease. Some of the most devastating, most stubbornly difficult diseases we face, cancer, developmental disorders, autoimmune conditions, neurological decline. When the regulatory machinery of the genome breaks down, the consequences can be profound, and understanding exactly how and why has become one of the central quests of modern biology. Let's start with cancer, because it's the most dramatic example. And honestly, once you understand gene regulation, cancer starts to make a terrible kind of sense. For a long time, people thought of cancer as this mysterious, almost random affliction, cells just going haywire for no clear reason. But what we now know is that cancer is, at its core, a disease of gene regulation. It's what happens when the controls we just talked about, all those switches and dimmers and loops and silencers, start to malfunction. Here's how to think about it. Your cells have genes that tell them to grow and divide. These are called proto-oncogenes, and they're completely normal, completely necessary. Every time you heal a wound, every time your bone marrow produces new blood cells, proto-oncogenes are doing their job. They're the accelerator pedal. And then you have another set of genes called tumor suppressors. These are the brakes. They tell cells when to stop dividing, when to pause and repair damaged DNA, and when to self-destruct if the damage is too severe, a process called apoptosis, which is this elegant, controlled cellular suicide that keeps damaged cells from accumulating. Now, in a healthy cell, the accelerator and the brakes are in balance. The regulatory machinery we've been talking about ensures that growth signals are expressed when they're needed and suppressed when they're not. But imagine what happens if a mutation hits a proto-oncogene and turns it into an oncogene, a version that's stuck in the on position. Suddenly you've got a cell with the accelerator jammed to the floor. Or imagine a mutation knocks out a tumor suppressor gene. Now you've lost the brakes. Either way, you get uncontrolled cell division, and if you get both, accelerator stuck and brakes gone, that's when things get really dangerous. The thing is, it's usually not just one mutation. This is something that took researchers decades to work out. Cancer typically requires multiple hits, multiple regulatory failures accumulating in the same cell lineage over time. A landmark paper by Bert Vogelstein and Eric Ferrin in 1990 laid this out beautifully for colorectal cancer. They showed that the progression from normal colon tissue to a benign polyp to an invasive carcinoma followed a specific sequence of genetic changes. First, a loss of the APC tumor suppressor, then activation of the KRAS oncogene, then loss of other suppressors like SMAD4 and TP53. Each step removed another layer of control. Each step made the cells a little more reckless, a little more autonomous. It was like watching someone systematically disable every safety system on a complex machine, one by one, until the thing was running completely out of control. And here's where the epigenetic regulation we talked about earlier becomes really important. Because it turns out that you don't always need a mutation, an actual change in the DNA sequence, to disable a tumor suppressor. Sometimes the gene is perfectly intact, but it's been silenced by aberrant DNA methylation. The cell has slapped methyl groups onto the promoter region of a critical tumor suppressor, and now that gene is locked in the off position even though nothing is wrong with the gene itself. This is called epigenetic silencing, and it's been found in virtually every type of cancer. The BRCA1 gene, which you may have heard of in connection with breast and ovarian cancer, in some tumors, it's not mutated at all. It's just been methylated into silence. Think about what that means. The information is still there in the DNA. The gene is intact. But the regulatory machinery has gone wrong, and the result is just as catastrophic. as if the gene had been deleted entirely. It's like having a perfectly good fire extinguisher bolted to the wall, but someone has painted over it so thoroughly that nobody can find it when the fire starts. And this actually opens up a really interesting therapeutic angle, because unlike mutations, epigenetic changes are potentially reversible. If a tumor suppressor has been silenced by methylation rather than destroyed by mutation, you might be able to reactivate it. There are drugs called DNMT inhibitors, DNA methyltransferase inhibitors, that can strip away some of those methyl groups and wake silenced genes back up. Azacitidine and decitabine are two examples that are actually used clinically, particularly for certain blood cancers like myelodysplastic syndromes. They're not magic bullets, and they're not precisely targeted. They affect methylation broadly, which creates its own problems. But the principle is remarkable. You're not killing the cancer cells directly. You're trying to restore their own internal regulatory controls. You're trying to give them back their breaks. But cancer isn't the only story here. Let's talk about something that might seem completely different, but is actually deeply connected: development. How a single fertilized egg becomes a fully formed human being with 220 or so distinct cell types, all carrying the same DNA but expressing completely different sets of genes. This is where gene regulation really shows off, because the developmental program of a complex organism is essentially a cascade of regulatory decisions. At each step, cells are receiving signals from neighboring cells, from gradients of signaling molecules called morphogens, from their position in the embryo. And these signals activate transcription factors that turn on specific sets of genes, which produce proteins that in turn influence the next round of decisions. It's a branching tree of regulatory choices, and once a cell goes down a particular branch, epigenetic mechanisms lock that decision in place. The cell and all its descendants remember what they've become. The classic example here involves a family of genes called the Hox genes, and their story is genuinely one of the most beautiful in all of biology. In the late 1970s and 1980s, researchers studying fruit flies, Drosophila, our old friend from the Morgan lab, discovered that certain mutations could cause bizarre transformations. A fly might grow legs where its antennae should be, or develop an extra pair of wings. These were called homeotic mutations, and they revealed that there were master regulatory genes controlling the identity of body segments. Edward Lewis, Christian Nusslein-Volhard, and Eric Wieschaus shared the 1995 Nobel Prize for working out the genetic control of early embryonic development, and the Hox genes were central to that story. But here's what's really wild. When researchers started looking at other organisms, mice, frogs, humans, they found that we have Hox genes too. Not just similar genes, but clearly the same genes, conserved across hundreds of millions of years of evolution. The Hox genes in a mouse are arranged along the chromosome in the same order as the body segments they control, from head to tail. And the regulatory logic is recognizably the same as in a fruit fly. A fruit fly and a mouse last shared a common ancestor something like 600 million years ago, and yet the fundamental system for patterning the body axis has been preserved. The machinery works so well and is so deeply integrated into the developmental program that evolution has kept it essentially intact across that incomprehensible span of time. And when Hox gene regulation goes wrong in humans, the results can be severe. Mutations in Hox genes or their regulatory regions have been linked to limb malformations, certain cancers, and developmental syndromes. There's a condition called synpolydactyly extra or fused fingers and toes, that's caused by mutations in the HOXD13 gene. The gene itself is a regulatory switch, and when the switch is broken, the downstream developmental program produces the wrong output. Now, here's something that connects back to what we were saying about the non-coding genome. Many of the regulatory elements that control Hox gene expression are located far from the genes themselves, sometimes hundreds of thousands of base pairs away. They're enhancers. Exactly the kind we talked about, looping through three-dimensional space to contact their target promoters. And some of the most consequential mutations in human disease aren't in genes at all. They're in these distant regulatory elements. A study published in 2014 found that a large proportion of the genetic variants associated with common diseases through genome-wide association studies, these massive surveys that scan the genomes of thousands of people, fall in non-coding regulatory regions, not in the genes themselves, in the switches that control them. This was a huge shift in how geneticists think about disease. For decades, the focus had been on finding mutations in protein-coding genes, the 1.5%. But it turns out that a lot of the genetic variation that makes one person more susceptible to diabetes, or heart disease, or schizophrenia, or autoimmune conditions lives in the regulatory architecture. A slightly different enhancer sequence might mean a gene is expressed at 90% of its normal level instead of 100, or expressed in a slightly different set of cells, or turned on a few hours later during development. These are subtle changes, not dramatic on-off switches, but over a lifetime, or across the critical window of embryonic development, they can matter enormously. And this is actually one of the reasons why the Human Genome Project, completed in 2003, didn't immediately deliver on its most ambitious promises. When the sequence was first published, there was enormous excitement, and rightly so, because it was one of the greatest scientific achievements in history. But the hope that we would quickly be able to read the genome like a blueprint and understand disease was, in hindsight, naive. Because having the sequence is like having the text of an extraordinarily complex book, written in a language you only partly understand. The protein-coding genes were the easiest part to interpret. They follow the codon rules that Nierenberg and Khorana worked out. But the regulatory grammar, the syntax that determines when and where and how much each gene is expressed, that's vastly more complex, and we're still learning to read it. Which brings us to something that changed everything about how we study gene regulation, and really about how we study biology in general. Because in the early 2000s, a set of tools began to emerge that gave researchers the ability not just to read the genome, but to edit it, precisely, cheaply, and in virtually any organism. And the most transformative of those tools has a name you've almost certainly heard, even if you're not sure exactly what it does. It's called CRISPR, and the story of how it went from an obscure curiosity in the genomes of bacteria to the most powerful tool in molecular biology is one of those tales that reminds you why science is so much stranger and more wonderful than fiction. It starts in the late 1980s in the coastal city of Alicante, Spain. A young microbiologist named Francisco Mojica was studying archaea. Those are the single-celled organisms that thrive in extreme environments, and the ones Mojica was interested in lived in salt marshes so briny that almost nothing else could survive. He was sequencing parts of their genome, tedious work at the time, and he noticed something odd. There were stretches of DNA that contained short, palindromic repeats, sequences that read roughly the same forward and backward, and these repeats were separated by unique spacer sequences. The pattern was unmistakable. Repeat, spacer, repeat, spacer, repeat, spacer, over and over. And Mojica thought, that's weird. What is that? Nobody knew, and for a while, nobody particularly cared. Mojica kept finding these strange repeat spacer arrays in other microorganisms too, not just his salt-loving archaea, but in all sorts of bacteria. By the early 2000s, he'd compiled a growing catalog of these sequences, and he proposed the acronym CRISPR, clustered regularly interspaced short palindromic repeats, which is a mouthful, but it stuck. The question was still, what are they for? Why would bacteria carry these oddly structured sequences in their genomes? And here's where Mojica had his breakthrough moment. Around 2003, he started comparing the spacer sequences, the unique bits between the repeats, against databases of known DNA. And what he found gave him chills. The spacers matched the genomes of bacteriophages. Phages, viruses that attack bacteria. Think about what that means. These bacteria were carrying, in their own DNA, little snippets of viral genomes. It was like finding a most wanted poster tucked inside the bacterium's filing cabinet. Mojica realized that this was an immune system, not an immune system like ours, with antibodies and white blood cells, but a genetic immune system. When a bacterium survived a viral attack, it could grab a small piece of the invading virus's DNA and store it in its own genome, filed away between those palindromic repeats. And then, if that same virus, or one closely related, ever came back, the bacterium could recognize it and fight it off. This was a genuinely revolutionary idea. Bacteria having adaptive immunity. It seemed almost too elegant. Mojica submitted his paper in 2003, and it was rejected. Repeatedly. Journal after journal turned it down. One reviewer said the findings weren't sufficiently novel. It wasn't published until 2005, and even then, in a relatively modest journal. Meanwhile, a French researcher named Gilles Vergniaud and a team led by Alexandre Bolotin were independently reaching similar conclusions. Science has a way of converging like that. When the data is there, multiple people start seeing the same pattern. But knowing that CRISPR was a bacterial immune system was just the beginning. The real question became, how does it actually work? How does a bacterium use those stored viral snippets to defend itself? That's where a cascade of discoveries in the late 2000s filled in the mechanism, and it's beautifully simple once you see it. Here's how it works. When a virus injects its DNA into a bacterium, special proteins associated with the CRISPR system, called Cas proteins, short for CRISPR-associated, grab a piece of the viral DNA and stitch it into the bacterium's CRISPR array as a new spacer. That's the acquisition step, the filing away of the mugshot. Then, the bacterium transcribes those spacers into short RNA molecules called CRISPR RNAs, or cRRNAs. Each cRRNA carries the sequence of one particular viral snippet. And here's the clever part. Those cRRNAs team up with Cas proteins to form a surveillance complex. The cRRNA acts as a guide. It floats around inside the cell, and if it encounters a piece of DNA that matches its sequence, meaning the same virus has come back, it binds to it. The Cas protein then cuts the viral DNA, destroying it. It's search and destroy, guided by a genetic memory. Now, there are different types of CRISPR systems. The one that changed everything for biotechnology was type 2, and specifically a protein called Cas9, found in the bacterium Streptococcus pyogenes. What made Cas9 special was its simplicity. In many CRISPR systems, the surveillance complex involves multiple proteins working together. Cas9 does it all by itself. One protein, guided by one RNA molecule, finds a specific DNA sequence and cuts both strands. Clean, precise, programmable. And this is where two scientists enter the story whose work would reshape modern biology. Jennifer Doudna, a biochemist at UC Berkeley, and Emmanuel Charpentier, a microbiologist who was then at Umeå University in Sweden. Charpentier had been studying the CRISPR system in Streptococcus pyogenes and had identified a crucial component, a small RNA molecule called tracer RNA that was essential for processing the guide RNAs. She and Doudna began collaborating, and in June 2012, they published a landmark paper in the journal Science. They showed that the Cas9 protein, guided by a single synthetic RNA molecule, they'd fused the cRRNA and TRA-cRRNA into one piece, which they called a single guide RNA, could be directed to cut any DNA sequence you wanted. Any sequence. You just had to design the right guide RNA, about 20 nucleotides long, and Cas9 would find the matching spot in the genome and make a double-strand break right there. Think about what that means for a second. For decades, molecular biologists had dreamed of being able to edit genes the way you edit text in a document. Go to a specific location, delete something, insert something, change a letter. There had been earlier tools, zinc finger nucleuses and talons, that could do targeted cutting, but they were expensive, time-consuming to design, and finicky. CRISPR-Cas9 was like going from hand-carving each letter in stone to having a word processor. You could design a new guide, RNA, in a day, order it for a few dollars, and have your experiment running by the end of the week. The barrier to entry collapsed. Within months of the Doudna-Charpentier paper, Feng Zhang at the Broad Institute and George Church at Harvard independently demonstrated that CRISPR-Cas9 worked in human cells. That was January 2013, and once that was shown, the floodgates opened. Labs around the world started using CRISPR to knock out genes, insert new sequences, correct mutations, in mice, in zebrafish, in plants, in fungi, in human cell lines. The technology spread faster than almost any tool in the history of biology. And here's what makes it so powerful for understanding gene regulation, which is really the thread we've been following. Remember all those regulatory regions we talked about? Enhancers, silencers, insulators, those stretches of non-coding DNA that control when and where genes are expressed. Before CRISPR, studying them was painstaking. You could correlate a regulatory region with gene expression, but proving that a specific enhancer was necessary for a specific gene in a specific tissue was incredibly difficult. With CRISPR, you could simply delete the enhancer and see what happened. Or you could make a single nucleotide change in a transcription factor binding site and ask, does this one letter matter? And the answer, often, was yes. Suddenly, the regulatory grammar of the genome became experimentally accessible in a way it never had been before. But CRISPR didn't just let you cut DNA. Researchers quickly engineered modified versions of Cas9 that could do other things. There's a version called dead Cas9, or dCas9, where the cutting ability has been disabled, but the protein can still find and bind to a specific sequence. You can attach other molecules to dCas9, an activator that turns a gene on, a repressor that turns it off, a fluorescent tag that lets you see where a particular sequence is located inside a living cell. There's base editing, developed by David Liu's lab at Harvard, where a modified Cas9 can change a single DNA letter, say converting a C to a T, without making a double-strand break at all. And there's prime editing, an even more refined version that can make virtually any small edit with remarkable precision. The medical implications started becoming real almost immediately. Sickle cell disease, which is caused by a single nucleotide change in the gene for beta-globin. 1A switched to a T, which changes one amino acid in the hemoglobin protein and causes red blood cells to deform into that characteristic sickle shape, became one of the first targets. Researchers realized they could use CRISPR to reactivate fetal hemoglobin, a version of hemoglobin that's normally switched off after birth. The idea was elegant. You don't even need to fix the mutated gene. Instead, you disrupt the regulatory element that silences fetal hemoglobin in adult cells. With fetal hemoglobin flowing again, it compensates for the defective adult version. In December 2023, the FDA approved Casgevy, the first CRISPR-based therapy for sickle cell disease and transfusion-dependent beta thalassemia. Patients' bone marrow stem cells are removed, edited with CRISPR to knock out that silencer, and then infused back. Early results have been remarkable. Patients who had been having regular pain crises, who had been dependent on blood transfusions, suddenly free of symptoms. Doudna and Charpentier shared the Nobel Prize in Chemistry in 2020, and it was notably one of the fastest Nobel recognitions in modern history, just 8 years from their key paper to the prize. The significance was that clear. But with power comes complexity, and with CRISPR, some of the hardest questions aren't technical, they're ethical. Because if you can edit the DNA in a person's bone marrow cells to treat sickle cell disease, those changes affect only that patient. They're somatic edits. They don't get passed on to the next generation. But what if you edit an embryo? What if you change the DNA in a fertilized egg, or in the germline cells that produce sperm and eggs? Those changes would be heritable. They would ripple forward through every subsequent generation. In November 2018, a Chinese biophysicist named He Jiankui announced that he had done exactly that. He had used CRISPR to edit the genomes of twin girls, Lulu and Nana, as embryos, attempting to disable a gene called CCR5 to make them resistant to HIV infection. The scientific community's reaction was immediate and overwhelmingly negative. Not because germline editing is inherently impossible to justify. There are thoughtful people who believe it could eventually be appropriate for devastating genetic diseases with no other treatment, but because He Jiankui's experiment was reckless. The medical need was questionable. There are effective ways to prevent HIV transmission that don't involve editing embryos. The informed consent process was deeply flawed. The editing itself was imprecise. The girls appear to carry mosaic mutations, meaning different cells in their bodies may have different edits, and the long-term consequences are unknown. He Jiankui was sentenced to 3 years in prison by a Chinese court in 2019. The He Jiankui affair crystallized a conversation that had been simmering since the moment CRISPR's power became apparent. Where do you draw the line? Editing somatic cells to cure disease. Most people are comfortable with that, at least in principle. Editing embryos to prevent severe genetic disorders like Huntington's disease, where a child would otherwise face certain neurodegeneration, that's harder. But many ethicists see a case for it, with proper safeguards. But what about editing for traits that aren't diseases at all? Height, intelligence, athletic ability? The term designer babies gets thrown around, and while the reality is far more complicated than that phrase suggests, most complex traits involve hundreds or thousands of genetic variants, each with tiny effects, and we're nowhere near being able to engineer them. The principle matters. The question of what we should do with this technology is going to be one of the defining ethical conversations of this century. And it's worth pausing here to appreciate something about the arc of this whole story. Because the journey from Miescher scraping pus off surgical bandages in 1869 to CRISPR-edited patients walking out of hospitals in 2024, free of diseases that had shadowed their entire lives, that's barely 150 years. In the sweep of human history, that's nothing. And yet, the depth of understanding we've gained in that time is staggering. We went from not even knowing what genes were made of to being able to rewrite them letter by letter in living human cells. But there's one more piece of this story that I think ties everything together, and it has to do with something we touched on earlier, the question of what the genome actually is. Because for a long time, we thought of it as a kind of static blueprint. You inherit your DNA, it contains your genes, and those genes determine your traits. But everything we've talked about, gene regulation, epigenetics, the vast regulatory landscape of non-coding DNA, the way environment and experience can alter gene expression without changing the sequence, all of that paints a very different picture. The genome isn't a blueprint, it's something more like a living language, a language that's constantly being read, interpreted, spoken aloud in some passages and whispered in others, with entire chapters left silent depending on who's doing the reading and when and where. Think about that metaphor for a second, because I think it captures something important that a blueprint never could. A blueprint is fixed. You draw it once, and then you build the thing it describes. But a language is dynamic. It has grammar and syntax, sure, but it also has context. The same word can mean completely different things depending on the sentence it's in, the tone of voice, the conversation that came before. And that's so much closer to how your genome actually works. Every cell in your body carries the same DNA, the same 3 billion or so base pairs, the same roughly 20,000 protein-coding genes, the same vast stretches of regulatory sequence. And yet a neuron in your brain looks nothing like a cell lining your stomach, which looks nothing like a red blood cell, which looks nothing like the photoreceptor cells in your retina that were letting you see the world today before you closed your eyes. Same script. Radically different performances. And that's because the genome isn't just a parts list. It's a score, and the cell is the orchestra. And the music that emerges depends on which instruments are playing, how loudly, in what combination, at what tempo. The conductor is the sum total of every signal the cell is receiving, from its neighbors, from the bloodstream, from the environment, from its own history. And the result is something far more fluid and responsive than any static blueprint could ever be. Here's what I find quietly astonishing about this. When the Human Genome Project was completed in 2003, there was this expectation. You could almost feel it in the press coverage, that once we had the sequence, we'd basically have the answer key. We'd understand genetic disease. We'd know why one person gets cancer and another doesn't. We'd crack the code of human biology. And in some ways, the project delivered on that promise magnificently. It gave us the reference sequence, the map, the foundation that every subsequent discovery has been built on. But in other ways, it revealed just how much we didn't understand. Because knowing the sequence of letters is not the same as understanding the language. It's like having every word in a novel laid out in front of you, but not knowing the grammar, the idioms, the cultural references, the way certain passages echo others from 200 pages earlier. The sequence was the beginning, not the end. And so much of what's happened in the 2 decades since has been about learning to read that language more deeply. Projects like ENCODE, the Encyclopedia of DNA Elements, spent years cataloging the functional elements in the genome, the regulatory switches and enhancers and promoters, all that non-coding territory we talked about earlier. And what they found was that something like 80% of the genome showed some kind of biochemical activity. Now, there's been a lot of debate about what that actually means. Biochemical activity isn't the same as biological function, and some researchers have argued that a lot of that activity might be noise, the molecular equivalent of background chatter. But even the most conservative estimates suggest that the functional portion of the genome extends far, far beyond the protein coding genes themselves. The regulatory architecture is enormous, and we're still mapping it. And then there's the whole world of what happens after transcription. We talked about how a gene gets transcribed into messenger RNA and how that RNA gets translated into protein. But even that description, as detailed as it was, is a simplification. Because in eukaryotic cells, cells like yours, the initial RNA transcript gets processed in all sorts of ways before it ever becomes a functional message. Sections get spliced out, other sections get rearranged, chemical modifications get added to the ends. And here's the thing that's really wild. Through a process called alternative splicing, a single gene can give rise to multiple different proteins. The cell can mix and match the segments of the RNA transcript, including some and excluding others, to produce different versions of the protein depending on what's needed. So that figure of 20,000 protein-coding genes? The actual number of distinct proteins your body can produce is vastly larger. Some estimates put it at over 100,000, maybe several 100,000, from 20,000 genes. The genome is not a simple one-gene, one-protein system. It's more like a jazz ensemble, riffing on themes, improvising variations, creating complexity from a surprisingly compact set of raw materials. And I think that's one of the most beautiful things about this whole story. The elegance isn't in the brute force of having more genes. It's in the sophistication of the regulatory systems that extract so much more from the genes you have. Bacteria might have a few thousand genes and relatively straightforward regulation. The ELAC-operon model we talked about, operators and repressors and inducers. But you, lying there right now, you're running a regulatory network of almost incomprehensible complexity. Transcription factors binding to enhancers that might be hundreds of thousands of base pairs away from the gene they regulate. Chromatin remodeling that opens and closes access to entire regions of the genome. Epigenetic marks that can persist through cell divisions, giving cells a kind of memory. Non-coding RNAs that fine-tune expression levels with extraordinary precision. Layer upon layer upon layer of control, all operating simultaneously, all interacting with each other, all responsive to signals from inside and outside the cell. And yet, somehow it works. Most of the time, it works remarkably well. You started as a single fertilized cell, and from that one cell, through billions of precisely orchestrated divisions, you built a body with trillions of cells organized into hundreds of distinct cell types, forming tissues and organs and systems that function together in ways that would make the most sophisticated engineering project look simple by comparison. And the instructions for all of that, not just the parts list, but the timing, the spatial organization, the feedback loops, the error correction mechanisms, all of it is encoded, directly or indirectly, in that long polymer of nucleotides that Friedrich Miescher first pulled out of white blood cells in a castle kitchen in Tübingen over 150 years ago. He had no idea what he'd found. He couldn't have. He called it nuclein, and thought it might have something to do with nutrition. Decades would pass before anyone connected it to heredity, more decades before its structure was solved, more still before the code was cracked, the regulation understood, the editing tools developed. And the story isn't over. It's not even close to over. Right now, tonight, while you're drifting off, there are researchers in labs around the world working on the next chapter. Some of them are developing new CRISPR variants with even greater precision. Others are studying the three-dimensional organization of the genome inside the nucleus, how DNA folds and loops in ways that bring distant regulatory elements into physical contact with the genes they control, creating a kind of spatial grammar on top of the linear sequence. Others are exploring the role of the microbiome, the trillions of bacteria living in and on your body, in influencing gene expression. Others are building synthetic genomes from scratch, writing DNA sequences that have never existed in nature, pushing the boundaries of what biology can do. And some of them are doing something that I think Miescher and Levine and Chargaff and Franklin and Crick and all the others would find absolutely magical. They're using DNA itself as a data storage medium. Because DNA is, when you think about it, an extraordinarily efficient way to store information. It's stable, it's compact. You could theoretically store all the data ever created by humanity in a volume of DNA that would fit in the back of a pickup truck. And we already have the molecular machinery to read it and copy it. Researchers have successfully encoded books, images, even video into synthetic DNA and read it back out again. It's not practical yet for everyday use. The writing and reading processes are still too slow and expensive, but the principle works. The molecule that life chose to store its own instructions turns out to be useful for storing ours too. There's something poetically satisfying about that. But honestly, for tonight, I don't want to leave you with the technology. I want to leave you with something simpler, something you can carry with you into sleep. Think about the fact that you are right now a living expression of DNA's four-letter alphabet. Every breath you're taking involves proteins encoded by your genes, built by ribosomes reading messenger RNA, regulated by networks of transcription factors and enhancers and epigenetic marks that have been fine-tuned by billions of years of evolution. The hemoglobin carrying oxygen to your brain right now, the molecule we talked about, the one that sickle cell disease distorts with a single amino acid change, the one that CRISPR therapies are now targeting. That hemoglobin is being produced at this very moment by cells in your bone marrow, reading the beta-globin gene on chromosome 11, following instructions that have been passed down, copied and recopied with astonishing fidelity through an unbroken chain of cell divisions stretching back to the very first organisms that figured out how to bind oxygen. You are connected through your DNA to every living thing on this planet. The same genetic code, the same codon table, the same amino acids, the same basic molecular machinery operates in you and in the bacteria, in the soil and in the trees outside your window, and in the whales in the deep ocean. That universality, that shared language of life, is one of the most profound things science has ever revealed, and it all started with a molecule, a long, twisted, elegantly simple molecule made of sugar, phosphate, and 4 nitrogen-containing bases. Adenine pairs with thymine. Guanine pairs with cytosine. 2 strands wind around each other in a double helix. And from those pairings, from that structure, from the information encoded in the sequence of those bases, everything follows. The storage of genetic information, the faithful copying of that information from one generation to the next, the reading of genes into proteins, the regulation of which genes are read and when and where, the development of a single cell into a complex organism, the evolution of life itself over 4 billion years, the ability now to read and edit and even write the code. It's all there, in the helix. It was always there. We just had to learn how to see it. So let that settle over you. Let the details blur if they want to. the names and dates and molecular mechanisms. What matters is the shape of the story, the long, patient human effort to understand the most fundamental molecule of life, the mistakes and the breakthroughs, the people who spent their careers chasing something they couldn't quite see, the quiet moments in labs late at night when something finally clicked, the way each discovery opened up 10 new questions, and how that's not a frustration but a gift, because it means there's always more to learn, always another layer to uncover, always another reason to be curious about the world you live in and the body you inhabit. You're made of DNA's handiwork. Every cell, every tissue, every thought forming in your mind right now as you drift closer to sleep. And the molecule doesn't rest either, somewhere in you. Right now, a helicase is unwinding a double helix, a polymerase is laying down new nucleotides, a ribosome is ratcheting along a strand of messenger RNA, building a protein one amino acid at a time. The machinery of life, running quietly, faithfully, while you rest. It doesn't need your attention. It doesn't need you to be awake. It just runs, the way it's been running since long before there were humans to wonder about it. The way it'll keep running long after tonight is over and forgotten. So let it run. Let it do its work. And let yourself sleep.